Hello everyone. Today we are moving to discuss about a new topic, monocot and dicot embryo. In this video we are discussing the structure and development of monocot and dicot embryo. Before going to the topic, we know that the plants are divided into seeded and non-seeded plants. In seeded plants there are two categories, that is gymnosperms and angiosperms, which means the gymnosperms and angiosperms both are having seeds. Okay, so what is the difference between the gymnosperms and the angiosperms? It is about their flowering characters. Gymnosperms are non-flowering plants and angiosperms are flowering plants. In angiosperms, which are the most advanced group of plants, and they are having the flowers, and they are characterized by two groups, that is monocots and dicots. So in this video we are discussing how the monocot and dicot embryo developing, and of course their structure also. Embryogenesis. So what do you mean by embryogenesis? It is nothing the development of a zygote into an embryo, which is by predetermined series of cell divisions and cell differentiations. So what do you mean by cell division, and what is the difference from that of cell differentiation? Cell division, which means it is just the multiplication of cells, that is from a single cell it is multiplied into or divided into 2, then to 4, then to 8. All these are just merely the multiplication, that is known as the cell division. And what about cell differentiation? Which means the group of cells, they are having particular function. In that case it is termed as cell differentiation. So cell division means they don't have any functions. But the cell differentiation, they are having the functions. Which means some of the cells which is destined to form the flower tissues, some of them become epidermis, some of them become vascular tissues. Such kind of functions that is attributed in cell differentiation. Embryo. After the embryo formation, that will germinates and grows into an adult plant. In seed plants, the embryogenesis, which occurs within the embryo sac of the ovule. Okay, so once we study the structure of ovule, we know that the embryo sac, which is nothing, the female gametophyte. You can see the entire events in this picture. There is a flower. Within the flower, you can see the male and the female sex organs. The male sex organ, which is commonly known as the stamen, which consists of the anther and the filament. Within the anther, you can see there is a bilobed anther. Within the anther, there is microsporangium. So microsporangium, within the microsporangium there is a microspore mother cell that undergo meiosis, that is forming the pollen grains or the microspores. So what is the difference between the microspores and the pollen grain? Microspores, when the microspore attains the cell wall, it is termed as the pollen grains. So you can see microspore mother cell, that is where the microspore mother cell, that is within the microsporangium. Within the microsporangium there is microspore mother cells are there. This microspore mother cell which undergoing meiosis. So what is the difference between the meiosis and the mitosis? Meiosis means which is the reduction division. This microspore mother cell, which is a diploid tissue or a sporophytic tissue, when it is undergoing meiosis, which become the haploid or gametophytic tissue, which means the pollen grains or the microspores as a result of meiosis of microspore mother cells are haploid. Okay, so that the pollen grains, within the pollen grains which undergoing the gametogenesis, or which undergoing the division to form the generative cell and the tube cell. After that, it is liberated from the anther or the microsporangia. Okay, right. On the same hand, you can see that the female sex organ, that is known as the carpel, which is consisting of the stigma, antistyle and the ovary. Okay, within the ovary there is the ovule, and you can see that the ovule, within the ovule there is megaspore mother cell. That megaspore mother cell undergoing the same events that is in the case of anther, that is the meiosis and the mitosis. So then it becomes the embryo sac. Within the embryo sac, you know that the embryo sac, which is having the egg apparatus, antipodals and the secondary nucleus. That is the megagametophyte or the female gametophyte or the embryo sac. Okay, once the pollination. So what do you mean by the pollination? The liberated pollen grains that is transfer into the stigma of the female sex organ, that is known as the pollination. So once they landed on the stigma, they will reach into the ovary. Within the ovary, they reach into the ovule. Within the ovule, there is the embryo sac. Within the embryo sac, there be the egg apparatus. Within the egg apparatus, there is the egg nucleus, so that the fertilization will happen. So you can see that the double fertilization. So which is the characteristic feature of the angiosperms. What do you mean by double fertilization? Double fertilization means, you can see in this case the pollen grains having generative cell and the tube cell and the nuclei which undergoing division to form two sperms. Okay, these two sperms, one of the sperm or one of the gamete which will fuse with the egg nucleus and forming the zygote, and the other one that is fusing with the polar nuclei to form the endosperm, triploid endosperm. That will discuss in another class. Okay, so this double fertilization which will affect into the formation of zygote, the diploid zygote. Okay, from this zygote into the development into the embryo, that is known as, that series of events is known as the embryogenesis. This embryogenesis which will happen by the result of cell division as well as the cell differentiation. Then come to the, I told you there are two kinds of plants are there, angiosperm plants are there, that is dicots and the monocots. Okay, monocots, which means all the most of the herbal plants or most of the lower plants, they are monocots only, and dicots means all the trees and everything which will comprises the dicots, all the grasses, such kind of plants which is included in monocots. Okay, when come to the structure, you can see on the right hand, you can see that the embryo which is consisting of two major parts, that is the cotyledons and an embryonal axis. Okay, the embryonal axis which is having these parts, that is the plumule, epicotyle, the point of attachment of cotyledons, hypocotyle and the radicle. Generally, the embryo having the cotyledons and an axis. On the axis, there is the leaf-like cotyledons are arises. Okay, I think all of you are very familiar with the germination of tamarind seeds. You can see that the tamarind seeds, once we throw out the tamarind seed, it will germinate, and you can see the cotyledons and everything is very clear in the case of that tamarind germination. You can see the cotyledons as a the parts of that seed as such. After that it germinates. Okay, that you can, once you see the parts of seeds, that is actually the cotyledons. Okay, so generally the embryonal axis bear the cotyledons. The cotyledons are actually the leaf-like structures which is otherwise termed as embryonic leaves, which is attached to the embryonal axis. The embryonic shoot apex, which is termed as the plumule. In this case, the embryonal axis which bear the shoot apex, that is known as the plumule, which the plumules develops into the future shoot. And there is root apex, which is known as the radicle, and the radicle which develops into the future root. The part of axis above the radicle, which is termed as the hypocotyle, and below the plumule, which is known as the epicotyle, which you can see in this picture. So briefly, the embryo, there is having an embryonal axis and the cotyledons. With the embryonal axis bear the shoot apex known as the plumule and the root apex known as the radicle, and along with the hypocotyle and the epicotyle. You can see the position of the hypocotyle. Actually, the hypocotyle is a position, that is the part above the radicle, that is known as the hypocotyle, and the epicotyle, which is having just below the shoot apex, that is the plumule, that is known as the epicotyle. You can see on this right hand picture there. So these are the parts or the structure of a dicot embryo. And just we discussing about that development. The crucifer type means the crucifers are the one category of angiospermic plants, and the development, that is a typical example, that's why the crucifer type. And the first cell division, we know that the zygote, which is formed after the fertilization or after the fusion of male and the female gamete within the embryo sac, that is forming the zygote. This zygote which undergoing the first division, which is an asymmetrical division. What do you mean by asymmetrical division? You can see on the right hand picture, the zygote undergoing a division to form a large cell and a small cell, which means the cells are not equal in their structure. That is why they are termed as the asymmetrical. So that you can see there is a small apical cell or small terminal cell and a large basal cell. That is why the division is termed as the asymmetrical. The apical cell which divides further, and the basal cell again divides, both apical cell divides by the vertically and the basal cell undergoing transverse division and forming a four-celled structure, that is marked as a quadrant stage, that is known as the pro-embryo, and by the shape of the cell it is termed as T-shaped. You can see on the quadrant stage there, you can see four cells are there, the four cells, and the apical cell, you can see the vertical division, and the basal cell undergoing a transverse division. The upper three cells of the proembryo, out of the four cells, the upper three cells which divide further both by anticlinal and periclinal division to forming a 32-celled globular proembryo stage. Okay, we know that the first T-celled proembryo having three cells. Four cells. Out of the four cells, the upper three cells of the proembryo divides further both by transverse division and longitudinal division and forming a mass of 32-celled globular proembryo. And the development of various organs of embryo or organogenesis, the so-called radicle, plumule, everything else occurs in this 32-celled proembryo. The outermost layer of cells of the globular proembryo, which develops into a distinct layer called dermatogen and which forms the future epidermis. That is means that outermost layer is termed as the dermatogen, which becoming the future epidermis. The inner cell of the upper region develops into the cotyledons, and the plumule, while the lower cells develops into the radicle, which means the embryo having all the preliminary parts of a future plants. Okay, outermost cells that is becoming the epidermis, and the inner cells that is becoming all the plant parts such as the cotyledons, the shoot apex known as the plumule, and the root cells that is known as the radicle. The cotyledonary cells which divides rapidly than the plumule cell. Therefore, the embryo becoming the heart shape. You can see on the right hand picture, you can see the position of plumule, radicle and the cotyledons there, and the cotyledonary cells which develops or which cell divide rapidly, and so that which will cover the plumule or the stem tip, so that the embryo becoming the heart shape. When the cotyledons are completely formed, they enclose the plumule at the base. You can see on the right hand picture there is a heart shaped structure, and the both wings are known as the cotyledons. These are growing rapidly than the stem tip, so that which enclose the stem tip, and altogether it looks like a heart shaped structure there. The lowermost cell formed by the basal cell which does not contribute to embryo structure, which means out of the 4 cells, only 3 cells are contributing to the embryo structure, which may be undergoing few more divisions to form a filamentous structure known as the suspensor. So what is the function of suspensor? Which means which anchors the embryo into the embryo sac wall, so that it will get nutrient absorption from the surrounding tissues. So the function of the suspensor, it is nothing, it is just pushing of the embryo into the embryo sac wall and getting the nutrients from the surrounding tissues. And when come to the second category, that is the monocot plants, here the structure of the monocot embryo. So the monocot embryo, what is the basic difference? The first mono-di, which means the dicots having 2 cotyledons. I told you 2 cotyledons. The dicot embryo having these 2 cotyledons, and whereas the monocots having single large cotyledon which is known as the scutellum. Whereas in the case of dicot embryo, they are having 2 cotyledons. So here in the monocot embryo, the single large cotyledon is called as the scutellum. And here the plumule and the radicle both are lateral in position. They are positioned in sidewise. You can see the picture there, and the scutellum. You can see the scutellum and the radicle and the plumule, both of them are on sidewise position. The plumule, which is protected by a covering of leaf-like structure known as the coleoptile. Okay, the shoot apex or the plumule, which is protected by coleoptile, and the radicle, which is protected by a covering known as the coleorhiza. Apart from the coleorhiza, there is a protective cap that is known as the root cap. So we know that the shoot apex and root apex, that are the most sensitive parts which have to become the future shoot and root, so that they are protected in very such kind of manner, that is the shoot apex by the coleoptile and the root apex by the coleorhiza, and along with this coleorhiza there is another protective structure that is known as the radicle. And when come to the development, so you can see the grass type. What do you mean by why are you tell that it is grass type? Which means one of the representative or one of the typical representative of the monocots are the grasses. So that you can see the development as a grass type. As we told in the case of dicot embryo, the first division of the zygote, here it is also like oblique or asymmetrical, and they are transverse. As in dicot embryo, which will producing a small apical cell and a large basal cell. Both cells which divide against at right angles to the first division and forming a 4-celled proembryo, which is like that in dicots. Okay, so the 4-celled embryo and further division, which is at different planes. As a result, the formation of a 16-celled and 32-celled proembryos. And from the proembryo, the development of various organs occurs and organogenesis occurs in the 32-celled proembryo, which means the embryo or the proembryo at 32-celled, after that only the organogenesis occurs. What are the organs? Which are nothing, the shoot tip, the root tip, coleoptile, coleorhiza and everything that is known as the organogenesis. The cells of lateral apical region develops into a single cotyledon or the scutellum. And below the scutellum, there is plumule and the radicle are developing. The outer layer of cells which develops into the coleoptile, root cap and the coleorhiza, which means the most prominent structures like the shoot tip and the root tip, which are enclosed within the layers of coleoptile, root cap and the coleorhiza, which means the outer layers or the exposing layers which is developing the coleoptile, root cap and coleorhiza. Coleoptile which protects the shoot tip and the coleorhiza which protects the root tip. Coleorhiza as it is a protective structure for root which stays in soil, because roots are present in the soil, hence this coleorhiza stays in the soil. But coleoptile, which is the covering or which is the protective part of the shoot tip which emerges from the soil. Okay, so that is about the structure of monocot embryo. Okay, that is about the dicot and monocot embryo structure and the development. If you have any doubts, you can comment below. That is about the structure. Thank you.